to start to grow inappropriately or move inappropriately. We're going to begin talking about all of these different processes that occur, and when these processes occur, that's when our cells become transformed into tumor cells. What I have shown here is some data from the National Cancer Institute, and this is data actually from 2007. It takes a while to compile data such as this. And they were looking at some of the most common cancers in the United States and what would be the 5-year survival rate for these various cancers. So one of the most kind cancers that you can develop would be a non-melanoma skin cancer. 5-year survival rate of that is 93%. That's pretty good. Melanoma involves melanocytes, or the cells that produce pigment in your skin, and actually that's the 8th most common cancer in the United States. It also has a fairly high survival rate, about 93%. It's pretty good if it's caught early. Then you have other types of cancers. Prostate cancer has a very high survival rate. Lung cancer, not so much. I'll talk about that shortly. Breast cancer, also a pretty solid survival rate. Colon and rectal, not so much. The reason for that is oftentimes it's caught too late. Non-Hodgkin's lymphoma is a tumor in your B and T cells of your immune system. Not such a good rate of survival. Actually, one of the most dangerous tumors you can develop is pancreatic cancer. To this date, actually this is a little bit high. It's really about a 4% 5-year survival rate. One of the reasons why pancreatic cancer is so deadly is, again, it's a sneaky cancer. People don't know they have it until it is too far along, and then by that time, the tumor cells have metastasized, and I'll talk about that shortly. Tumor cells have metastasized and established other secondary tumors throughout the body. Another very bad cancer to develop is ovarian cancer for the same reason. Ovarian cancer is also a silent cancer. Oftentimes, the cancer is very advanced by the time the patient realizes that something is wrong. Okay? So always in the case of cancer, early detection is one of the keys to be able to treat a cancer successfully. Does anybody have a question? Okay. Certain cancers don't have a much better survival rate than they did back in 1930. So some cancers just are too difficult to treat. One would be lung cancer, pancreatic cancer, ovarian cancer. If you develop these, your chances of 5-year survival are not much better than they were back in 1930. Okay, almost 100 years ago. Not much better. They're very difficult cancers to treat, and I'm going to go over some of the reasons why through the course of the lecture. Okay? Does anybody have a question so far? All right. You might know that much of what I'm going to be talking about today is not in your book. Okay. All right. The most important thing for a cell to become a tumor cell is the cell has to sustain a series of mutations that will allow the cell to lose its normal gene expression program and just become a proliferating cell. And that's what tumors are. Tumors are actually cells that have lost their normal gene expression program, and they're just starting to proliferate. Usually, a tumor cell has to be able to proliferate enough to where the tumor contains about 2 to the 11th cells. 2 to the 11th cells. That's a lot of cells. That's before you can feel it. Before you can feel it, the tumor is that far along. And so what are the genetic changes? What are the kinds of genetic changes that have to occur for a cell to become transformed? Well, most of the genetic changes some of these proteins that activate the cell cycle, and also proteins that regulate the cell cycle. All of these different critical proteins that control a cell's proliferation rate, those things have to undergo mutation before a cell loses its control. And so one of the ways that the genetic changes that can actually occur in the genes that encode these proteins are overexpression. And so all of these genes that encode proteins that activate the cell cycle, genes that encode proteins that activate the cell cycle, those are called proto-oncogenes. Genes that encode proteins that activate the cell cycle to push the cell into a proliferative mode, those are called proto-oncogenes. Because if the gene that if one of these proto-oncogenes actually sustains a mutation, then the proto-oncogene becomes an oncogene. And oncogenes are encoding proteins, abnormal proteins, that push the cell into a proliferative mode. So the kinds of genetic changes that can turn a proto-oncogene into an oncogene are shown here. The first one is one of the things I just mentioned a minute ago, and that's overexpression. Whether or not the promoter, if there's a translocation, where the proto-oncogene is translocated into a new promoter that makes that gene encode vast amounts of the protein that's going to push the cell into the cell cycle, or if there's an amplification of the proto-oncogene, so that again you have vast amounts of a protein that is going to push the cell into a proliferative mode. And again, that's what tumor cells do. They proliferate. Another thing that can happen is that you will have, for example, a mutation within the gene that encodes the proto-oncogene. And when that mutation occurs, it might occur in a region of the gene that encodes a critical domain in the protein. So if there's a point mutation in a critical domain of the protein, that means the protein is not going to be responsive to mechanisms to turn it off. And I'm going to give you some wonderful examples of this shortly here. So again, point mutations that result in a protein not being able to regulate itself and turn itself off. And also, you can have, that's what I just described, you can also have a point mutation within a promoter that keeps the proto-oncogene transcribing, and again, excessive amounts of the protein. So in 3 of these mechanisms shown here, what you end up with are excess amounts of a protein that pushes the cell into the cell cycle. That's gene amplification, that is translocation, where the proto-oncogene is put under control of a new promoter that causes the gene to be transcribed continuously, or a point mutation within the promoter of the proto-oncogene such that it's transcribed continuously. All of these result in abnormal amounts of protein in the cell, pushing the cell into a proliferative mode, and then, as I just described, you have a point mutation within the coding sequence of the gene that results in a protein that just will not be turned off or regulated. And I'm going to give you some examples of that. Okay? Can I repeat anything or explain anything further? Are we okay so far? Okay. All right. One of the first examples of translocation is shown here. People who develop chronic myelogenous leukemia. Chronic myelogenous leukemia actually comes from a translocation that occurs in the cells that are going to become that are going to become white blood cells. So all of our blood cells actually start out with a stem cell that's present in our bone marrow. All of our blood cells start out as stem cells in the bone marrow. And what happens in chronic myelogenous leukemia is that there's a stem cell that actually sustains what is called a translocation, where a segment of chromosome 22 is carried over to chromosome 9, and that segment from chromosome 9 is then carried on over to chromosome 22. It's called a reciprocal translocation because the chromosomes actually exchange fragments. And in this case, you get a translocated chromosome 9 and a translocated chromosome 22. What was interesting is where the translocation occurs. On chromosome 9, you have a cytoplasmic tyrosine kinase called c-Abl. And on chromosome 22, you have a segment, it's called the breakpoint cluster region, or BCR. And always in this translocation that occurs in chronic myelogenous leukemia, what happens is the gene encoding the c-Abl tyrosine kinase is translocated to a region in this breakpoint cluster on chromosome 22, and that becomes and that's going to develop into the BCR-Abl gene, where the point where the translocation occurs forms what is called the BCR-Abl gene. And this translocated chromosome is actually called the Philadelphia chromosome because it was first identified at the Wistar Institute here in Philadelphia. This is the responsible protein. It's an oncoprotein. It's a fusion protein, not normally one that the body makes, because it's a result of a translocation. And it produces what is called the BCR-Abl fusion protein. It overstimulates cell division. It's a protein kinase. It phosphorylates many substrates within the cell that cause stem cells that should differentiate into white blood cells. Instead, those stem cells just start to divide, and they take over. They actually take over the blood cell population of the patient. And it's called chronic myelogenous leukemia. This is actually treatable. This is actually treatable with a drug called Gleevec, G-L-E-E-V-E-C, Gleevec. It's treatable. Gleevec was only developed in the last decade or so. Prior to that, these patients would suffer over time with their leukemia. It was called smoldering leukemia. It had a term called smoldering leukemia. Eventually, the patients would die. Now, you have Gleevec, so the patients can be treated. Gleevec doesn't cure the leukemia. It's a treatment. Because what Gleevec does is a small molecule that actually binds to the BCR-Abl kinase protein and prevents it from phosphorylating its cellular targets. And these patients have to take Gleevec every day in order to prevent the c-Abl, BCR-Abl kinase from inappropriately phosphorylating its targets and activating cellular proliferation. Anybody in here know how much Gleevec costs a cancer patient every month? $8,000. $8,000. If you don't have insurance, what are you going to do? You're going to die. Isn't that terrible?
Isn't the medicine that costs the United States $8,000 like a long-term week or something, but it's cheaper for cancer?
That's not cheap. No, Gleevec still is $8,000 a month. Yeah. It's impressive in its ability to treat leukemia. but it's not impressive what it's going to do to your life if you don't have insurance. Okay, question?
How much is the cost of produce?
That's why it costs so much, because Novartis Pharmaceuticals that developed it took a long time developing it and had to pay a lot of people, paid a lot of clinical trials, and by the time they were able to put it into the market, that's how much it was going to cost for them to be able to profit from their effort. It's a sticky wicket. That's all we can say. All right, let's move on. Okay. So first I talked about how you can have translocation. Translocation is an example of what happens with chronic myelogenous leukemia. In this next example, what I'm going to do is I'm going to talk about how tyrosine kinase receptors, if a portion of the gene that encodes a tyrosine kinase receptor is deleted, then what's going to happen is the tyrosine kinase receptor is going to remain constantly on and firing into the cell to activate the cell cycle. And that's what we have here. This is a simple diagram of the normal epidermal growth factor receptor. It's a transmembrane protein. It actually has a ligand binding site, if you can remember from our cell signaling lecture, has a ligand binding site where it binds to epidermal growth factor. When the epidermal growth factor receptor binds to epidermal growth factor, what happens is it activates its tyrosine kinase domain. And the tyrosine kinase domain, what it does is it phosphorylates all kinds of proteins that are in the cytoplasm, where it phosphorylates these various signaling proteins on tyrosine residues, hence it's called the tyrosine kinase. And what's been found in a number of different types of tumors is that the gene for the epidermal growth factor receptor has sustained a mutation. And the component of the gene that actually encodes the epidermal growth factor binding domain or the ligand binding domain has been deleted. And so what we're looking at here is not so much a point mutation within the gene that encodes the epidermal growth factor receptor, but an entire deletion of the gene that actually encodes the ligand binding domain. And so what you're left with is a truncated tyrosine kinase receptor that is constantly signaling into the cell. It doesn't need to bind to epidermal growth factor to do that. It remains constantly active. It was first identified and called the ERB protein. And this particular mutated tyrosine kinase is found to be active in a whole series of different types of tumors, including lung cancer, bladder cancer, breast cancer. A lot of different cancers have unfortunately been established by mutation of the epidermal growth factor receptor, where the ligand binding domain is actually deleted from the gene, so that the proto-oncogene, which is the normal gene for the normal epidermal growth factor receptor, then becomes an oncogene. And it's the ERB oncogene that generates this ERB protein that is really the mutated form of the normal receptor. Can I repeat anything or explain anything further? Are we okay? So what does this thing do again? It activates the phosphorylation cascade in the cell and activates the cell cycle. And again, that's what tumors do. They have inappropriately activated cell cycles that allows for the tumor cell to just continue to proliferate. They lose their normal gene expression program. The cells no longer have a normal structure and function. Instead, they simply proliferate. Okay. So I've talked to you a little bit about some of the proteins that are involved activating the cell cycle. Also, what I want to mention is that, remember these checkpoint proteins that we talked about when we were talking about the cell cycle, the checkpoint proteins that, for example, retinoblastoma protein that prevents the cell from going past the G1 checkpoint into S phase unless the cell is ready. There's another checkpoint protein that's involved in making sure that the cell has plenty of proteins necessary for making the mitotic spindle. Also, the M checkpoint is that wonderful anaphase, that metaphase checkpoint where all the kinetochore microtubules have to be attached to kinetochores on chromosomes. If one of these checkpoint proteins fails, that's another way for a tumor cell to develop. So not only can you actually have a mutation in a proto-oncogene to turn it into an oncogene, and these would be proteins that activate the cell cycle. But if you have a failure of your checkpoint proteins, that will also allow for the cell cycle to continue to march through. Okay? So you have two different mechanisms going here. First, you have a mutation that will turn a proto-oncogene into an oncogene. And again, proto-oncogenes encode proteins that activate the cell cycle, such as tyrosine kinase receptors, for example. And then another thing that you will have for a tumor to develop is a failure of your checkpoint proteins to stop the cell cycle and make sure that the cell is healthy and should be proliferating. Question?
I remember you said in one of the last lectures when you were explaining this that if your checkpoints don't work, the cell can go back to EMAT.
The question is, if the checkpoint doesn't work, the cell will go back to GNOT. No. When the checkpoint protein is working, then the cell will go back to GNOT. Okay. If this checkpoint protein isn't working, whether the cell's ready or not, it's just going to march on through. And that's one of the things that will distinguish a tumor cell from a normal cell, is that the tumor cell will have abnormal numbers of chromosomes, it will have translocated chromosomes, it will have deleted chromosomes, all kinds of chromosomal abnormalities will be found in tumor cells because checkpoint proteins are working